So welcome to part 2 of bacteriology MCQs. So we start right through. Number 31. Gastrointestinal infections due to mucosal invasion are caused by all the following pathogens, except, so the question here is asking about mucosal invasion. So we have Shigella, Clostridium perfringens, Campylobacter, enteroinvasive E. coli, Helicobacter pylori. So it can't be D because from the word enteroinvasive E. coli, EIEC, so it's definitely not that. Then it can't also be H. pylori because we know H. pylori invades the gastric mucosa, causing ulcers. Okay. Campylobacter jejuni, it also causes dysentery or bloody diarrhea via invasion of the mucosa. Shigella too. Shigella also causes dysentery. So the answer here is Clostridium perfringens. So how does Clostridium perfringens cause disease? Because it causes food poisoning, but how does it cause food poisoning?
Does it produce like a toxin?
Yes, so it's toxin-mediated food poisoning. So the exact toxin is an enterotoxin. Okay, so there is no mucosal invasion. Good. Number 32. Pseudomonas aeruginosa's biological characteristics include the following except. Okay, so these are things you have to be knowing. It's quite unfortunate because you have to be knowing for each and every bacteria. Okay, so there are some mnemonics that are found in the internet where there are oxidase positive bacteria, lactose fermenters. Remember I had said Citrobacter, Enterobacteriaceae, Escherichia coli, and Klebsiella. There's also the catalase positive organisms. So you can use those mnemonics because they can help you both in sport, SAQ, and MCQs. Okay. So number 1, non-lactose fermenter. So the lactose fermenters are Citrobacter, Enterobacter, E. coli, and Klebsiella. So that is true. Oxidase positive, that is very true. That one you have to remember. Pseudomonas is oxidase positive. Ferments maltose. No, it is a non-lactose and non-maltose fermenter. Okay, so that's the answer here. Production of pigments, that is true. What are the names of the pigments that Pseudomonas produces? Who knows?
There's pyoverdin.
Which is color? Green.
Yeah. Then pyruvin, pyomelanin, and is it cyan or something like that?
Thiocyanin, which is the blue one. Yes. So those are the pigments that Pseudomonas produces. And then non-fastidious gram-negative bacilli. That is true. Meaning it doesn't require nutritional requirements or nutritional conditions. Okay. So the following are small gram-negative coccobacilli of clinical importance in human beings, except. So how to answer this question? First of all, if you've had a bacteria, okay, here, all these bacteria, you have heard of them. And you have to remember your classifications. If you can remember, gram-positive bacteria, we started with Coccus and Bacillus. Coccus, we had 2, Staphylococcus and Streptococcus. Bacillus, we had the Clostridium and Bacillus. And then we had the filamentous, which are the actinomycetes and Nocardia. Okay, then you go to gram-negative. The same way we are classifying. So that is something that by now you should have it in on your palms, okay? Because it is a classification that you have to know. Okay. Then we go to gram-negative bacteria. We have the cocci, the bacillus, and the curved ones. For the cocci, We had Neisseria, where we had Neisseria meningitidis, Neisseria gonorrhea. For the bacillus, we had Haemophilus influenzae. We had, trying to remember the others, Brucella. We had Bordetella pertussis. We had, which other one? Pasturella, Francisella. Okay. Then for the curved ones, we had, those are the Coccobacilli. For the curved, these are the Vibrio, Campylobacter, Helicobacter. Then the bacilli, which are the common ones that we know, lactose fermenters and non-lactose fermenters, E. coli, Klebsiella, Enterobacter, Citrobacter. Then for the non-lactose fermenters, Pseudomonas, Salmonella, Shigella, Yersinia. Okay, so you should be having that in your fingertips. Okay, so that it gets easy for you. Now for this question, they've asked for gram-negative. And we know that Streptococcus pyogenes is a gram-positive bacteria. So that becomes the answer. Okay, there are charts where you're going to know. There are charts in the internet where they classify these bacteria. So please go through them. Okay. So the answer here is D. I hope everyone got this question correct. Number 34. A 47-year-old woman presents with difficulty swallowing, double vision, and descending flaccid paralysis after eating home canned food. The toxin causing this condition acts by. So first of all, from this presentation, what do you think this patient has?
Botulism.
Botulism, very good. So there are symptoms of botulism, and then there's eating canned food, so home canned food. So we are thinking, so the person got food poisoning and it was from a toxin. So the question is, how does this botulism toxin act? So what's the answer?
A.
A, good. Blocking acetylcholine release at the neuromuscular junction. Very good. That is for Clostridium botulinum neurotoxin. So some words, they may use that. Some may use it cleaves the snare proteins. Okay. So if you see something like snare protein, think of Clostridium botulinum. Okay. Then blocking glycine and GABA release. What does that? What does choice B?
Tetani. Clostridium tetani.
Clostridium tetani. Good, because it's going to cause the paralysis. We call it spastic paralysis. Then choice C, stimulating CAMP production. This is, yeah. So they are the main two. Which are they? You start with Vibrio. And the other one? Which other one? One of the E. coli. Was it the heat labile or the heat stable? Which one stimulates CAMP and which one stimulates CGMP? Do you remember the chart I sent you? Yeah? Who remembers? So the mnemonic was labile in the air, stable in the ground. So stimulating CAMP, which is A, there's this letter A, okay? The letter A here in CAMP is for heat labile E. coli, enterotoxigenic E. coli, okay? ETEC. Then for heat stable, it will be stimulating cyclic GMP. Okay, cleaving IgA. What are the three bacteria that produce IgA protease?
Haemophilus.
Haemophilus, good.
You have Streptococcus.
Which Streptococcus?
Is it pyogenes?
pneumoniae. Okay.
Okay.
Yeah, pneumoniae. And then the last one? Neisseria. Okay. So both Neisseria meningitidis and Neisseria gonorrhea. Depolymerizing actin filaments. Which bacteria does that?
Is it syphilis? I mean, the one that causes syphilis. The
Treponema?
yeah.
No. Did we do with you guys Shigella? Or have you read it somewhere? I want you to go check on it. Check on Shigella, okay? So I want you to go check on mechanism of, so apart from the Shiga toxin, that is, the Shiga toxin, do you remember the Shiga toxin? How does the Shiga toxin work? It inhibits protein synthesis, okay? Yeah, so I want you to go check on that. But here, it was a very direct question. The answer was A. Okay. A 5-year-old boy presents to the casualty department the history of dog bites. Okay. On examination, the site of the bite was swollen, red and hot. Gram staining, the culture reveals gram-negative coccobacilli. Okay. Which is the most likely organism? So, dog bite. So, first of all, before you even answer this question, you remember the classification. So, the coccobacillus are Haemophilus, Pasteurella, Francisella, Yersinia. Is Yersinia coccobacillus? It cannot be actinomyces. First of all, actinomyces is gram-positive. So choice E is wrong. Is Yersinia coccobacillus? What is the shape of Yersinia?
A bacillus.
It's a bacillus. So choice D is wrong because it's not a coccobacillus. Actinomyces is wrong because it's not gram-negative. Now you're left with A, B, C, and D. Sorry, A, B, and C. Then from here you start reasoning. Let's start with choice A, Haemophilus. Haemophilus, how do we get Haemophilus? How do you get Haemophilus?
Air droplets.
Air droplets. So that is definitely wrong. Then we're left with Pasteurella and Francisella. So you might maybe confuse and you don't know. What you should remember is Francisella is not spread by dogs. It is spread mostly by, it is zoonotic, yes, but it's spread mostly by rabbits. That's how I usually remember for Francisella, rabbits. Then Pasteurella, dogs and cat bites. Okay. Pasteurella multicoda. Okay. So you see how we have eliminated. So don't rush. You have to get the content well, then analyze from the choices. Pasteurella, what's the question? Pasteurella multicoda, okay. The commonest clinical manifestation is pharyngitis. So it cannot be pharyngitis. Pharyngitis means, remember when you hear the word itis, it means inflammation. Then this is inflammation of the pharynx. So this is the pharynx. So it can't be that. That if it was pharyngitis, it will be more of an upper respiratory tract infection, things like Haemophilus. Contains metachromatic granules. Metachromatic granules, where did we hear that? Where did we get across that?
The corynebacteria bacteria.
Corynebacteria, very good. You remember, seen on Albert stain, the metachromatic granules or the Loeffler media. Okay. So it's not that. Non-capsulated. Is it capsulated? So Pasteurella is capsulated. Dermonecrotic toxin is only produced by high virulence strains. So that is false. It is produced by both high virulent and low virulent strains, and then gram-negative Coccobacillus. So from your classification, you would have already got the answer without even reading the other choices, because we already know Pasteurella is among the gram-negative Coccobacilli. 37. Chances of detection of Treponema pallidum in a specimen is lowest in. Okay. Now, so before we answer this question, we have to remember there are 3 stages of syphilis. Okay. We had the primary syphilis, secondary syphilis, and tertiary syphilis. Then remember on the side, we also had the latent syphilis. Let's start with primary syphilis. Primary syphilis, what was the characteristic of primary syphilis? What, if you hear what, you'll know that this is primary syphilis.
A painless primary canker.
Yes, a canker, a painless ulcer. It can be on the penis, it can be on the labia, on the reproductive parts of the reproductive system. Okay. Now, when you go and scrape there, or you get an exudate from that painless ulcer, it is going to be very high in Treponema pallidum spirochetes. Okay. You remember we do dark microscopy where we see light spirochetes on a dark background. Okay. So that you're going to have high levels of the spirochetes. Now let's go to the secondary stage. Now, in secondary stage, what are we expecting in secondary stage? What will you hear on when we talk about secondary stage? Steve has been quiet. Are you there?
I think secondary stage is that dissemination.
Dissemination, very good. So that means that there is systemic spread of the spirochetes. It has now gone to other parts of the body, to the skin, causing the rashes, to the mouth, causing mucocutaneous lesions, to the lymph nodes, causing lymphadenopathy. Okay, so at this stage, the spirochetes are all over the body. So do you think that there'll be high levels of spirochetes when you go to when you scrap ulcers?
No.
No, it makes sense, isn't it? Because the spirochetes have disseminated to all parts of the body. Okay, now let's go to tertiary syphilis. So tertiary syphilis or the late stage syphilis, what happens?
This is the neurosyphilis.
Neurosyphilis. So it has gone and affected the brain and the spinal cord. So here is where you have the neurological symptoms. Okay. And then here, when you do CSF analysis, you're going to see the spiral sheets. So, and then definitely you also have congenital syphilis, not really a stage, but one of the, we can classify it as a stage, but it is only seen in newborns. So what happens is if the mother had active syphilis, it passes through the placenta, then the child gets the congenital syphilis. In newborns, when you take samples of their blood, you're going to see high levels of Treponema spirochetes. Okay, so the key takeaway is ulcer scrapping from secondary stage syphilis has the lowest number of spirochetes, but remember, serology will still show the spirochetes are present. Okay, so things like VDRL, the rapid antigen test, the RPR, okay, they will still show high levels of syphilis. So they'll be positive. Okay. Are we clear? So that's why choice A is wrong, because at the blood level, you're still going to see the levels of the syphilis, the spirochetes. So are we clear there? Number 38. False statements on potential human pathogen in the genus Leptospira. Okay. So the Leptospira grows in artificial culture media. That is true. Susceptible to penicillin. That is true. Which are the bacteria that are not susceptible to penicillin? So first of all, you have to ask yourself, how does penicillin work? Yes.
Yeah, it acts on the bacterial cell wall.
On the bacterial cell wall, isn't it? It inhibits synthesis of the cell wall, binding to the, you remember the name, penicillin binding proteins. Okay, they are found on the peptidoglycan layer. So from that, which bacteria are not susceptible to penicillin?
Mycoplasms.
So yes, so bacteria that have no cell wall, like Mycoplasma, Ureaplasma, and all those. Okay. Now, is Leptospira there? No, Leptospira has a peptidoglycan layer as a cell wall, so it will be susceptible to penicillin. It's associated with damp areas populated by rodents. That is true because that is the common source. Is a serotype of L. biflexa. So you see for Leptospira, okay, let's go to choice E first. May be detected in urine of an infected person by lab tests. That is actually how we detect it. Okay. L. biflexa is a different type of serotype. It is then an infectious form. Okay. So they are not the same. Those are two different types of bacteria. Okay. A false statement on elementary bodies of Chlamydia
Nathan, I have a
trachomatis.
question.
Yes, sorry.
So I know choice B is wrong specifically for Leptospira, but let's say we get a bacteria like, I don't know, Streptococcus, like one which is known to have resistance against penicillin.
Like Staph aureus.
Yeah, you know, something like that. But you also have another choice that's also right.
So
Like,
you go with the other choice. You know, the thing is, if you see, like, for example, the following is true about Staph aureus, there are some species of Staph aureus that are susceptible to penicillin, but they are not common, you see, because, you know, we have methicillin resistant Staph aureus. So if you see a better answer, it's better you go with that. The other one, that is.
okay, thank you.
Okay, yeah, because the most Staph aureus species are resistant to penicillin and the other products. So penicillin, methicillin, and those. Okay. Yeah. So a false statement on elementary bodies of Chlamydia trachomatis. So I think I had explained this. You remember them? Elementary bodies? Do you remember? And the reticulate bodies?
Yes.
So care to explain it to us?
Okay, so elementary bodies is how it's formed when the bacteria enters, and then then it's going to replicate in the RE cells, forming reticulate bodies, and these are the ones that are seen when these form inclusion bodies that are seen when we stain the bacteria.
So in summary, elementary bodies are the extracellular ones, okay? And they are the infectious ones. Then the reticular bodies are the ones that are intracellular. They're found inside the host. Now, if you know that, you now start asking yourself, if elementary bodies are extracellular, they are going to be the infectious form. And if they're the infectious form, will they be metabolically active or metabolically inert?
Should
Active.
be active.
Elementary bodies, they are found outside. Listen, they are found outside and they are the infectious. They cannot be metabolically active because the metabolically active are the ones that are going to be dividing. And those are the reticulate bodies. Okay. Okay, so reason it like this. The elementary bodies are the extracellular ones and are the infectious ones. They have already divided and they are just, their work is just to infect cells. But the reticulate bodies are found inside the hosts and they are the ones that are metabolically active. What that means is they are rapidly dividing and they are non-infectious. It's like a baby. The reticulate bodies are the mother forming the baby. Then the elementary bodies are the babies. Now they are done. They're just going to infect. So they have already multiplied inside the cell and then they're going to burst, forming the elementary bodies. So a false statement can withstand harsh environmental conditions. That is true because they're outside of the body. If you're outside the body, you're going to withstand harsh metabolic conditions, environmental conditions. But reticulate bodies are going to be less resistant to these environmental conditions. Okay? And then are metabolically inert. That is true. Spore-like, when they mean spore-like, they mean that they are resistant to things like heat, chemicals, and harsh temperatures. So that is true. Are they infectious form? Are they the infectious form? That is true. Okay. And then can be visualized inside the host cell. That is false. Those are the reticulate bodies. Okay. Are we clear? So if you once you understand how they are, it gets easier. In summary, the elementary bodies are the extracellular ones and they are the infectious form. They are already outside the cell. So they're going to infect other cells. Reticulate bodies are the ones that are inside the cell, can be visualized inside the host cell, and are the non-infectious form. And they're the ones that are replicating. Okay. So I hope that's clear. Typical pneumonia is caused by, so when we talk about typical versus atypical, when we talk about typical pneumonia, this means that it presents with the normal signs of pneumonia, the typical, okay? The common ones. For example, you will have a productive cough. In fact, that is the most common feature that you're going to differentiate between atypical pneumonia and atypical pneumonia. Atypical pneumonia, let me write it down because it's going to, there is atypical pneumonia, ATYPCAL, versus typical pneumonia. So in atypical pneumonia, you're going to have dry cough. Then the fever will not be exaggerated, just be a mild fever. But for typical pneumonia, which is the common type of pneumonia, you're going to have fever, high fevers up to at least 40 degrees. And then you're going to have productive sputum or a productive cough characterized by sputum. Now, the bacteria that don't sound, that have hard names, usually cause atypical pneumonia. That's how I usually remember. So things like Legionella pneumophiliae, Coxiella brunetti, Chlamydophila psittaci, Mycoplasma pneumoniae. Those are the ones that cause typical pneumonia. Then Haemophilus influenzae, that one causes atypical. No, it causes typical pneumonia. Then these ones cause atypical pneumonia. In fact, Mycoplasma pneumoniae is the most common cause of atypical pneumonia, leading to the dry cough. It's also called walking pneumonia. So in case you are asked Which bacteria causes walking pneumonia? The answer is Mycoplasma pneumoniae. Then Chlamydophila psittaci, this is the one that was associated with birds, pigeon droppings. Coxiella brunetti, this is the Q fever. Okay. Yeah. Yeah. What's that question? Scrub typhus is caused by so there, I mean, it's quite unfortunate because there's so many types of typhus and you have to remember them. There's epidemic typhus, there's this scrub typhus. Now let's explain. So here the answer is, what is the answer? What causes scrub typhus? What causes scrub typhus? Are you there?
I'm not so sure.
Okay, let's eliminate. Higher. We go to rickettsia. The most common rickettsia we know is Rickettsia prowazekii. Rickettsia prowazekii causes epidemic typhus caused by the lice. Okay, take note of that. It is lice that causes it. Okay. Okay. Let's go to Rickettsia typhi. Rickettsia typhi is caused by the flea. Okay. Flea. It causes endemic typhus. Clear? Then scrub typhus is caused by Orientia tsutsugamushi. Okay, this is caused by the mite. Should I repeat? I hope you know these pests.
Yes, please. Yes, please.
Let me start. Rickettsia typhi, this one, choice B, is spread by the flea. So if you have a table, just write Rickettsia typhi, flea. It causes endemic typhus. Okay. Then we go to epidemic typhus, which is Rickettsia prowazekii, spread by the lice. You know lice, the ones that are found in clothes. Okay, Rickettsia prowazekii, lice, causes epidemic typhus. Then we go to scrub typhus, Orientia tsutsugamushi, which is spread by the mites. Okay. Then you're going to ask yourself, Rickettsia, what causes it? It causes Rocky Mountain spotted fever, which is spread by tick. Okay, this one, Rickettsia, tick. This one, Conori, I don't know. So that is low yield. So are we clear? Can someone read back what I've said? So you tell us the disease and the pathogen. And sorry, the disease and what transmits it. Someone read. Let's start with Rickettsia typhi.
Endemic typhus, and it's transmitted by fleas.
Flea, good. Oriental tsutsugamushi.
Spread by mite.
So what does it cause? Scrub typhus. Okay. Rickettsia prowazekii.
It's lice.
Spread by
And
lice.
then
Causes?
epidemic type.
Typhus. Good. Then rickettsia.
Rocky Mountain
Spotted
and
fever.
then
Spread by
tick.
tick. Yeah. So sorry. There are so many insects that you have to know. Okay? You have to remember them because they are going to ask. Okay? They even on spot, they can put for you a tick. Then they ask you higher. Name 2 diseases spread by such an animal, such an insect. So you have to say rickettsia or Rocky Mountain spotted fever. Okay? Or they bring a louse, or something like that. Okay? So go through that. Higher. The following highly pathogenic obligate intracellular bacteria is transmitted by body louse. So we know this. What is the answer? We have just done this.
D.
Rickettsia prowazekii. It causes epidemic typhus. Higher. Brucella is transmitted through all the following except, you remember Brucella, which causes brucellosis? Inhalation of aerosols, that is true. Broken skin, true. Consumption of unpasteurized milk, that is true. Eating undercooked food, that is true. Person to person, I've never heard of that. So this was, I hope everyone got this correct. Yeah. We continue. The following require culture on cell lines, except now, if you were to look at all these bacteria, let's say even you didn't know what the question is asking. From all this, there is a common thing you're seeing. These are all bacteria, zoonotic bacteria, isn't it? Now, one thing I forgot to ask, all those rickettsias, Rickettsia typhi, prowazekii, tsutsugamushi, all those, what drug do we use?
Is it doxycycline?
Yes, so we use the tetracycline class, or most common first line is doxycycline. So don't forget that. Okay. So what do we talk about? What is a cell line? So cell line is where we culture an organisms that are obligate intracellular. Okay. So this question would have been phrased, the following are obligate intracellular organisms except. Now let's see. Coxiella is an obligate intracellular. Ehrlichia schaffensis is obligate intracellular. Orienta tsutsugamushi, obligate intracellular. Rickettsia africae, obligate intracellular. So the only one that is facultative intracellular is Bartonella henslea. Okay, it can be cultured on chocolate agar. What does Bartonella henslea cause? I think I had mentioned this. Even Steve had asked a question. What does it cause?
Cat scratch fever.
Yes, cat scratch disease, which is characterized by granulomatous inflammation. Okay. Number 45. Anaplasma phagocytal phylum. Yeah. So let us eliminate. Choice B is useful in treatment. So most likely this is a zoonotic disease, even if you have not heard of it. You see this bacteria that you have not heard of, most of them are zoonotic diseases. So if you are to see this, anaplasma, it sounds zoonotic. So drug of choice, doxycycline. Okay. Higher. Facultative gram-negative bacteria, most zoonotic diseases are obligate intracellular. So A is wrong. B is also wrong. Chocolate agar is the medium of choice. It cannot be because it is obligate intracellular. So you can't use that. Wrong. Transmitted by ambyloma. So you're left with choice D and E. Exists in two forms, small and large cell variant. And then choice E, transmitted by Amyloma americanum. That is false. Amyloma americanum transmits Echilichia schaffensis. So this one is transmitted by the tick. It's called Ixodes tick. So D is the correct answer. Even if you didn't know, you would have eliminated. Okay.
I have a question.
Yes.
Okay, so for choice A, it's written facultative gram-negative bacteria. How do you know it's not facultative anaerobic or facultative intracellular? Like, is that something you're just supposed to know based on the bacteria?
Yeah, so you see for these zoonotic bacteria, most of them are obligate intracellular. The rickettsias, things like Anaplasma, Echilichia schaffensis, all those. You see those, most of those zoonotic bacteria, those are obligate intracellular, meaning that they have to be inside the cell. You know, obligate means a must. Okay, so that is something you should know. The others, yeah, that's something you should know. But the good thing is they won't ask so many questions from like the ETCs of those bacteria. From Rickettsia to Anaplasma, they ask maybe 1 or 2 questions. Okay, so it's not a must you stress yourself. In fact, if you know all these typhus that we talked about and know that they are obligate intracellular, I mean, that's enough. And know that doxycycline is the mode of treatment. That's enough, honestly, because you can't start going now into each and every specific bacteria. Okay. Just know those typhus and the specific insects. Yeah, and doxycycline. That's enough. Okay.
Okay, thank you.
Yeah. Okay, can we take like a 2-minute break, then we come continue.
Okay.